Okay, so imagine trading this sort of blunt force of chemotherapy for something much more precise, almost like a scalpel, delivering powerful drugs exactly where they need to go inside the body.
Yeah, that's the promise, and it's driving a ton of research.
It really is. And today we're doing a deep dive into sources looking at a really cutting-edge approach using these things called nanoemulsions for targeted drug delivery, specifically against breast cancer cells.
Right, and these are incredibly tiny, think nanoscale droplets, maybe around 100 nanometers across, super small.
And the why here, the mission that the sources really highlight, is getting around the big problems with traditional treatments.
Exactly. Chemotherapy, well, we know the challenges. Systemic toxicity is a huge one. It hits healthy cells too. Then there are issues just getting enough drug to the tumor, bioavailability, and of course the big one, drug resistance. Cancer cells just fighting back.
Right, it feels like there are significant barriers there. And these nanomulsions, they're being presented as, well, a potential game changer, a way to boost precision, make the drugs work better.
And potentially make treatment easier on the body, more biocompatible, by guiding it straight to the target.
So how do they actually do that, the precision part?
Okay, so the sources lay out two main ways this works. First, there's passive targeting. It cleverly uses the tumor's own flaws against it.
Flaws, like what?
Tumors often have leaky blood vessels, and their drainage system, the lymphatics, doesn't work very well.
Ah, okay, so these tiny nanomulsions, because they're so small, they can sneak through the leaky vessel walls into the tumor.
Exactly. And then they get stuck there because the drainage is poor. They're small enough to get in, but find it harder to get out.
Got it. So the tumor environment itself kind of traps them.
Precisely. That's called the enhanced permeability and retention effect, the EPR effect. It's a well-known phenomenon in nanomedicine.
Okay, that makes sense. Passive. What's the second strategy?
That's active targeting. This adds another layer of, let's say, specificity.
And this means changing the surface of the nanomulsion somehow, adding something specific.
Yep. Think of it like sticking tiny keys, or tags scientists call them ligands, onto the outside of the nanomulsion.
Okay.
These keys are designed to fit specific LOX receptors that are often way more abundant on cancer cells than on normal healthy cells.
I see. So you combine that leaky vessel effect, the passive part, with these specific keys looking for cancer cell lock.
That's the idea. Combine passive and active targeting to really guide these drug-loaded nanoparticles right to the tumor, minimizing damage elsewhere.
It sounds incredibly elegant, but making them, getting these tiny complex droplets just right must be, well, really difficult.
Oh, it absolutely is. It's a major engineering challenge. The sources mention techniques like ultrasonication, phase inversion. It's complex stuff.
Right.
You need them to be the right size, stable enough to survive in the bloodstream, loaded with the drug effectively. It's not just mixing oil and water, it's controlling nanoscale physics and chemistry.
And what really struck me is that the sources suggest they're not just delivery vehicles, not just tiny trucks dropping off cargo. They actually interact with the tumor environment.
Yes, that's a fascinating angle. They can apparently influence signaling pathways within the tumor cells. They can help trigger apoptosis, programmed cell death.
Sort of telling the cancer cells to self-destruct.
Exactly. And they can also inhibit angiogenesis.
Which is stopping the tumor from growing new blood vessels to feed itself.
Right. So it's potentially a multi-pronged attack, not just delivering a poison. And this is particularly relevant for some tough-to-treat breast cancers, like triple-negative or HER2 positive types mentioned.
So pulling it all together, these nanomulsions look like a really advanced approach, trying to tackle those core limitations of older cancer therapies head-on.
Yeah, you look at the big picture, it feels like a significant step towards more effective, less toxic treatments. But, the sources are clear. There's still a way to go.
Right. The challenges. What are the big hurdles mentioned?
Well, clinical translation is always a big one. Taking promising lab results and proving they work safely and effectively in actual patients. That's a huge jump.
And I imagine manufacturing them, making these complex things consistently, affordably, and on a massive scale.
Definitely. Scalability is key. Plus, designing even smarter ones, multifunctional systems that maybe combine diagnosis and therapy, tailored for personalized medicine. That adds another layer of complexity.
Exactly. It's a long road from the lab bench to the clinic.
No doubt about it. Groundbreaking research is one thing, routine clinical reality is another.
So that brings us to a final thought for you listening. Thinking about this path from lab to clinic, what crucial step, what specific breakthrough do you think is absolutely essential next to really unlock the potential of this nanoemulsion technology for personalized cancer treatment? Something to mull over.